Photosynthesis and light. The sun is the source of energy for almost all life on Earth. Light energy from the sun is captured by plants and other photosynthetic organisms, and converted into stored chemical energy. The energy is stored in molecules such as glucose, which provide a source of food for organisms that cannot use light energy directly. Visible light is composed of a spectrum of colors, which can be separated using a prism. A prism bends rays of light and separates the colors because each one has a slightly different wavelength and is refracted to a slightly different degree. Visible light has a range of wavelengths, but the most important regions of the spectrum for photosynthesis are red and blue. The color of an object is determined by the wavelength of the light that it reflects back into our eyes. A blue shirt appears blue because it reflects blue light, which our eyes can perceive, but the shirt absorbs other wavelengths that fall on it and we do not see those colors. A black object absorbs all wavelengths of light, while something white reflects them all. Most plants have green leaves. This tells us that they do not absorb the green part of the spectrum. Green light is reflected and makes the leaf appear green. Looking closely at the structure of plant cells, we can see that the green color is due to the chloroplasts, which contain a green pigment called chlorophyll. Chlorophyll is unable to absorb green light, which it reflects, but it does absorb other wavelengths well. Red and blue light are absorbed particularly well and provide the energy needed for photosynthesis. The red and blue ends of the visible spectrum are the wavelengths that the photosynthetic pigments in plants absorb most efficiently. The rate of photosynthesis is highest when plants absorb these wavelengths.
Photosynthesis. Photosynthesis is a complex series of reactions catalyzed by a number of different enzymes. To aid understanding, we can consider photosynthesis in two stages called the light-independent and the light-dependent reaction. Both the light-dependent and the light-independent reactions take place in the chloroplasts of plant cells. The first stage is known as the light-dependent reactions because light is essential for them to occur. The light-dependent reaction occurs on the thylakoid membranes of the chloroplast and is powered by light energy from the sun. Each thylakoid is a flattened sac, so the space in the middle is narrow. The thylakoid membranes form stacks called grana, which may be joined together by intergranal membranes. Light is absorbed by photosynthetic pigments such as chlorophyll, which are found on the granal membranes. There are several pigments found in plants and each one absorbs light of a slightly different wavelength. The pigments are associated with proteins that are involved in electron transport, proton pumping and chemiosmosis. The photosynthetic pigments are combined into two complex groups, called photosystems I and II, which absorb the light energy and use this to boost electrons to a higher energy level so that they become excited. 1. The first step in the light-dependent reaction is the photoactivation of photosystem II. Pigment molecules in the photosystem absorb light energy and boost electrons in a molecule of chlorophyll to a higher energy level. The electrons are accepted by a carrier protein molecule at the start of the electron transport chain. 2. Photosystem II has to replace these lost electrons, and it does this by taking them from water. Water is split into electrons, protons and an oxygen atom. Since the splitting is brought about by light energy, it is called photolysis. The oxygen is released as an excretory product. 3. Excited electrons travel along the electron transport chain into photosystem I. As they do this, they lose energy, but this is used to pump protons into the thylakoid interior. The thylakoid interior is small and so a proton concentration gradient builds up quickly. The protons then flow out through a large channel protein, almost identical to the one in mitochondria, which contains the enzyme ATP synthase. This time, though, the formation of ATP is called photophosphorylation, and it occurs between photosystems II and I. 4. Absorption of light energy causes photoactivation in photosystem I, boosting more electrons to an even higher energy level. The electrons that arrive from photosystem II replace those that are displaced. The electrons at the higher energy level are combined with protons in the hydrogen carrier NADP+ to form an ADPH+. H+, the two products of the light-dependent reaction, ATP and NADPH+, H+, are used to drive the light-independent reaction. Cyclic and non-cyclic photophosphorylation. When ATP is produced using energy from excited electrons flowing from photosystem 2 through photosynthesis 1 and onto NADP+. The process is called non-cyclic photophosphorylation. When light is not a limiting factor, the light-independent reactions may proceed more slowly than the light-dependent reaction, so that the supply of NADP+ runs out. This means the electrons boosted up from photosystem 1 have no acceptor available to take them. They rejoin the electron transport chain near the start and generate more ATP for the light-independent reaction. This alternative pathway is called cyclic photophosphorylation.
Light-independent reactions. ATP and hydrogen ions and electrons are used in the second stage of photosynthesis, the light-independent reactions. During the light-independent reactions, carbon dioxide, taken in from the air, is combined with hydrogen and ATP to form a range of organic molecules for the plant. The conversion of inorganic carbon dioxide to organic molecules such as glucose is known as carbonization. ATP provides the energy for the process. The series of reactions that occurs during photosynthesis is summarized as carbon dioxide plus water converted into glucose plus oxygen. The light-independent reaction occurs in the stroma of the chloroplast and is a cyclic pathway called the Calvin cycle. ATP and NADPH plus H plus formed during the light-dependent stage supply energy and reducing power for the Calvin cycle. The final product of the cycle is carbohydrate. As this is a cycle, what goes in must leave, so 3 carbons enter in 3 molecules of carbon dioxide and 3 carbons leave in 1 molecule of triose phosphate, which can be used to form glucose or other organic compounds. 1. At the start of the cycle, the acceptor molecule ribulose bisphosphate combines with incoming carbon dioxide from the air to form glycerate 3-phosphate. This reaction is called carbon fixation. It is catalyzed by RUBP carboxylase, an enzyme that is sometimes called rubisco. 2. The ATP and NADPH plus H plus from the light-dependent reaction convert the glycerate 3-phosphate into triose phosphate. Glycerate 3-phosphate therefore becomes reduced to triose phosphate. No more phosphate is added, so the only input from ATP is energy. 3. 6 molecules of triose phosphate are produced, but only 5 are needed to reform the ribulose bisphosphate to keep the cycle going. The extra triose phosphate leaves the cycle, and since it takes a phosphate with it, this is replaced in the cycle from ATP. 6 turns of the Calvin cycle produces 2 triose phosphate molecules, which can be combined to form the final product, glucose. Some triose phosphate molecules will follow other pathways to make other organic carbohydrate molecules, such as sucrose or cellulose, or other molecules that the plant needs, such as amino acids, fatty acids, or vitamins.
The action spectrum and the absorption spectrum, and how the rate of photosynthesis is measured. The spectrum of colors that humans can perceive is just part of the electromagnetic spectrum. The range of wavelengths in visible light is from 400 nm to about 700 nm. Light provides the energy for photosynthesis, but not all wavelengths are absorbed equally by the chloroplasts. If white light is shown onto chloroplasts, the wavelengths they absorb depend on the pigments present in them. When photosynthesis occurs, carbon dioxide is used and oxygen is released. The mass of the plant will also increase as glucose is used to produce other plant materials. Any of these 3 factors can be used to measure how quickly the reactions of photosynthesis are occurring. Rapid oxygen production indicates a high rate of photosynthesis. If the rate is measured for a range of wavelengths, a graph known as an action spectrum can be built up. Chlorophyll is more sensitive to some wavelengths of light than others and produces excited electrons at the highest rate at these wavelengths. The more excited electrons are produced, the faster the rate of photosynthesis. A typical action spectrum shows that blue and red light are used most efficiently. Absorption and action spectra vary for different species of plant. Some plants contain different pigments such as carotene and xanthophylls, which absorb wavelengths that chlorophyll cannot. These pigments, known as accessory pigments, absorb some yellow and green light and account for the photosynthesis that occurs in this range. Aquatic plants release bubbles of oxygen as they photosynthesize, and if the volume of these bubbles is measured for a period of time, the rate of photosynthesis can be determined directly. Aquatic plants also remove carbon dioxide from their environment, causing the pH of the water to rise. Carbon dioxide dissolves in water to form a weak acid, so as it is removed, the pH will go up. Therefore, another way of determining the rate of photosynthesis experimentally is to monitor the change in pH of the water surrounding an aquatic plant over a period of time. Terrestrial plants also remove carbon dioxide from their surroundings, but this is difficult to measure. It can be done experimentally by supplying a confined plant with radioactive carbon dioxide, which can be measured as it is taken up and released from the plant. A third method of measuring the rate of photosynthesis in plants is to determine their biomass at different times. This is an indirect method. Samples of the plants can be collected and measured at different times, and the rate of increase in their biomass calculated to determine their rate of photosynthesis.
Limits to photosynthesis. The rate at which a plant can photosynthesize depends on factors in the environment that surrounds it. On a warm, sunny afternoon, photosynthesis will be more rapid than on a cool, shady morning. More oxygen will be produced and more carbon dioxide used. But photosynthesis cannot increase beyond certain limits. The effect of light, temperature, and carbon dioxide in the air can be measured experimentally, varying one factor while keeping the others the same. At low light intensities, there may be a shortage of the products of the light-dependent reaction, so photosynthesis is slow. At point A, the plant is saturated with light and cannot photosynthesize at a faster rate even with more light. This indicates that some other factor is limiting the reaction. Temperature affects enzymes that catalyze the reactions of photosynthesis, so that if the temperature is too low, the reactions may proceed very slowly. At low concentrations of carbon dioxide, there may be insufficient carbon dioxide to be fixed in the Calvin cycle. At higher carbon dioxide concentrations, another factor may limit the reaction. The level of carbon dioxide in the atmosphere is relatively low, and carbon dioxide is frequently a limiting factor for photosynthesis. Horticulturalists increase the yield of their crops by maximizing the rate of photosynthesis in their glass houses. They do this by keeping the air inside warm with heaters, increasing the intensity of light using lamps, and, in some cases, increasing the concentration of carbon dioxide.